Alright, so here's cellular respiration video 2. This is about glycolysis. Okay, and we'll talk about why there's pictures of champagne and wine and yeast and stuff here too in a second. So this is that same screen we keep seeing. What's the point? The point is to make ATP. So this is going to seem incredibly complicated way to do that, but this is what we've evolved to do. Alright, so glycolysis is the very first step. We're trying to break down glucose. Now remember, we want to access all the energy in all of those bonds, and in order to do that, it involves a lot of steps. So, glycolysis, glycolysis. Glyco means sugar, lysis means to cut, so it's splitting sugar. So we're taking glucose, a 6-carbon sugar, and turning it into 2 molecules of pyruvate, which are 3 carbons each. And this is an ancient pathway. This is one of the original ways that prokaryotes also got energy. Okay, so this is where energy transfer first evolved, is glycolysis. As remember that prokaryotes did not have mitochondria. So the first part does not take place in the mitochondria. Alright, and this is the starting point for all of cellular respiration. But it's inefficient. Why? Because we're only generating 2 ATP at this step for every 1 molecule of glucose. Okay, and there's an investment of energy too. So this basic part, this original part that prokaryotes did too, the glycolysis, doesn't give us a whole lot of oomph for each thing that we're eating here. So the development of the mitochondria really gave us a ton more energy to work with. Right, so you're not going to be able to run an entire human being just through glycolysis. You're going to need all those other things that happen in the mitochondria to be able to develop a big complex organism like that. Alright, so the soccer player says, that's not enough ATP for me. Definitely no way are we going to run a complex organism like a human being on just that little bit of ATP. So this occurs in the cytosol of the cell. So as I said, why is it occurring there? This is one of the ancient ways that that cells were getting energy. So this is was developed in the prokaryotes, and of course there was no mitochondria in a prokaryote. Right, so I always give away the answers of the penguin. I always forget the penguin is coming. So I just gave you that answer. But that would make a nice little mini open response question or something. That's a good thing to think about. Okay, giving you that evolutionary perspective in a little more detail. Prokaryotes. Okay, the first cells, no organelles. They lived in an anaerobic atmosphere. There was no oxygen. Life on Earth originally evolved. Oops, come on, pointer. There you go. Without any free oxygen in the atmosphere. So energy had to be captured from organic molecules in the absence of oxygen. So we're doing the part of cell respiration that doesn't require any oxygen. So prokaryotes that evolved glycolysis are ancestors of all modern life. So the enzymes that are used in glycolysis are similar among all organisms, and the genes that code for them are highly conserved. So most of the organisms on Earth are going to have the same enzymes for doing glycolysis. And so we can compare eukaryotes, bacteria, and archaebacteria using glycolysis enzymes. So bacteria were developed 3.5 billion years ago, doing glycolysis in the cytosol, doesn't really require any organelles. Then oxygen came around in around 2.7 billion years ago, and that's when we started to have the photosynthetic bacteria and algae And eukaryotes developed around 1.5 billion years ago and started to have membrane-bound organelles. So these things kind of develop together. First you have no oxygen, and you have bacteria that can get a little bit of energy from organic molecules without any oxygen. Then as oxygen develops and, the photosynthetic bacteria and algae come around the same time as the oxygen, and then a billion years later you start getting eukaryotes. So all life, all organisms share a couple key processes. Okay, they all do protein synthesis, they all do glycolysis, and they all do DNA replication. So glycolysis is one of these core things that all living things on the planet do, because it was a part of the early life of prokaryotes. Okay, and the enzymes of glycolysis are well conserved, meaning that we see them across organisms. And here's a nifty one from the penguin. You mean we're related? Do I have to invite them over for the holidays? Of course you do. There was plenty of bacteria present for your holiday feasts, I'm sure. All right, so glycolysis is 10 reactions. And these 10 reactions in your book are on page 166 and 167. So we are converting glucose, 6 carbon, to 2 molecules of pyruvate, which each have 3 carbon. All right, so the first step, of course, involves some investment of ATP. We're going to use 2 ATP to convert glucose here into the fructose 1,6-biphosphate. All right, so it's going to take, it takes a couple steps in there, and we use 2 ATP to do that because we're ditching some phosphates in there to make this happen. Then we're going to take the fructose 1,6-biphosphate and divvy that up. Come on. Oh, we're using some fuel. Okay, into dihydroacetone phosphate and glyceraldehyde 3-phosphate. Right, so these each have 3 carbons and they've got phosphate attached to them. Oh, that gives you what we were just talking about. All right, so at this point we're done having to use ATP, and we start getting some ATP out of it too. Alright, so after a few more steps, we're going to end up with the pyruvate. Okay, and along the way, okay, here you're going to have a reaction converting the 2 NAD plus to 2 NADH. What kind of reaction is that? So that sugar here is getting oxidized and this is getting reduced. We've got a big redox reaction happening there. All right, and the 2 phosphates, okay, in and out, so that we end up, okay, being able to convert 4 ADP to 4 ATP. So we finally get some energy out of this. So glycolysis, okay, produces 4 ATP and 2 NADH and consumes 2 ATP. So our net yield, meaning how does it all balance out, is that we're gaining 2 ATP and 2 NADHs out of glycolysis. And that's the bottom line of what to know here, that we're starting with glycolysis, that we're ending with 2 molecules of pyruvate, okay, that our net yield is going to be 2 ATP and 2 NADH. All right, and we've got enzymes along every step of the way. And I don't think you have to memorize these. I've had to memorize these a few times in my life. I don't believe that you have to, but I will check to be sure of that, but I don't think so. Alright, so here's another way of looking at the glycolysis. Remember that glucose is a very, very stable molecule, and it needs some activation energy to break it apart. So we're going to phosphorylate it. That's why we're consuming some ATP in the beginning, because we're going to use that phosphate. We're going to phosphorylate the glucose. All right, so this is where we're using up 2 ATP. Then we're gaining 4 ATP and 2 NADH to end up with 2 pyruvate. Right, so glucose becomes 2 pyruvate and 2 water molecules. Here's our 2 ATP and our 2 NADHs and a little bit of hydrogen ions, 2 hydrogen ions. So there's the energy investment phase, which is an endergonic reaction. We're investing some ATP. We're phosphorylating that glucose. Lighting a match there. Okay, so minus 2 ATP. Then we go into our energy payoff phase, which is an exergonic reaction. We're harvesting a little ATP and a little NADH. Okay, so there's our 4 ATP. Right, and our NADH is like money in the bank because it's our electron carrier. And you're going to see how this plays out in the next part. Right, so there's our net yield. 2 pyruvates, 2 molecules of water, 2 ATP, 2 NADH, and 2 hydrogen ions. Okay, that's our net yield. And that's important for moving forward after glycolysis. So here's the first 5 reactions of glycolysis. So glucose enters the cell, and once it's inside the cell, hexokinase comes along and is like, hey, glucose, you need to be phosphorylated. And so that's what happens. That's why we're investing some ATP here, because ATP ditches its phosphate and glucose ends up with a phosphate. So using hexokinase is the enzyme that drives this phosphorylation reaction, giving glucose a phosphate. That's why glucose, the first step, goes from being glucose to being glucose 6-phosphate, because it's just a glucose with a phosphate on it. Then another enzyme comes along, phosphoglucose isomerase, and that one is going to change glucose 6-phosphate into its isomer, fructose 6-phosphate. Then we're going to use another, ATP is going to ditch another phosphate that it's ready to get rid of, being driven by this enzyme, the phosphofructokinase, so that we're adding another phosphate. So here now we've got this sugar, which is a fructose sugar with a phosphate on both ends. Then we're ready to split. Okay, now it's super unstable and it's ready to split using these 2 enzymes. So just to reiterate before we move on to the next part, glucose comes in the cell. Hexokinase says, ATP, give glucose a phosphate. So glucose, regular glucose, becomes glucose with a phosphate. Glucose 6-phosphate. What is that 6 about? Well, the phosphate is attaching to the 6th carbon. Then this enzyme, okay, you can tell by its name what it does, phosphoglucose isomerase. Phosphoglucose, we're taking our glucose with a phosphate on it and changing it into its isomer. So that's what an isomerase does. So it changes it into fructose 6-phosphate. And then phosphofructokinase, okay, again, a kinase, that comes up all the time when we're doing phosphorylation. Now we're going to have 2 phosphates on this fructose. So then the next is the actual split. Okay, so this is when glucose actually gets lysed, right? So this is the split, and we're creating 2 3-carbon sugars that are isomers of each other. So aldolase does the splitting, and then isomerase makes them into the 2 isomers. Okay, so that's it for the next slide. So, all right, moving forward, we're not really so interested in this 3-carbon sugar. We really want this one, so the isomerase drives the process that way, because only this 3-carbon sugar is what we're going to use. So after we split and we get these, the isomerase will change this one into that one, because we want this one moving forward. So the reason this one's circled in green is this is the 3-carbon sugar that we need. So we're going to have these 2 3-carbon sugars of this, okay? And that is going to drive this next part of the reaction, which begins kind of the happy part where we're getting some energy and getting stuff that we need. So first we're this is on page 167 of your book, I think, in a little bit better detail. This is where we have the redox reaction. The sugar is oxidized by the transfer of electrons and the hydrogen ion to the NAD plus forming NADH, the redox reaction. And that reaction is very exergonic, and the enzyme uses the released energy to attach a phosphate group to the oxidized substrate, making a product with a lot of potential energy. And that will take us on to the second half. Okay, so the second half of glycolysis is another 5 reactions. Alright, so this is that isomer change we were talking about earlier, that this is the one that we really want to be using, the G3P, this 3-carbon sugar with the phosphate on it. Okay, so we're going to be producing NADH. Alright, so the NAD plus becomes NADH. The G3P is donating the hydrogen, and we're oxidizing the sugar. So reducing NAD plus to get NADH. And now we're going to start getting some ATP out of this, right, as we continue to convert this 3-carbon sugar into pyruvate. Right, so PEP sugar is going to donate a phosphate. Okay, and that's that substrate-level phosphorylation. So we get some more ATP. So ADP becomes ATP. Okay, and they pay off, finally, some ATP. So the first 5 steps are about getting down to the 3-carbon sugars, and then the second half is converting the 3-carbon sugars into pyruvate, and along the way is when we get our ATP and our NADH. Okay, substrate-level phosphorylation, the last part. And the last steps of glycolysis, okay, where did the phosphate come to make ATP? Well, it came from the sugar substrate PEP. Okay, that last sugar, the phosphoenolpyruvate, the PEP, still is a 3-carbon sugar with a phosphate attached to it. So we've still carried these phosphates through from the phosphorylation reactions earlier. And so now we're going to take that phosphate and add it onto ADP to get ATP. And that leaves us with a 3-carbon sugar that doesn't have a phosphate on it. and those are the pyruvate. Okay, so there's the PEP, okay, and the enzyme. Let's show you that reaction. Okay, and so we're converting PEP to be a new molecule, pyruvate, a 3-carbon sugar, and a molecule of ATP because we ditched our phosphate. Remember that we like to pass that phosphate off as soon as possible. It's that bad boyfriend you want to get rid of. So now we've got pyruvate and some ATP. So the P transfers from PEP, that last 3-carbon sugar, to ADP. Okay, using a kinase enzyme. So we go from ADP to ATP. We get energy. Yeehaw. Right, so the phosphate came directly from the substrate. So energy accounting of glycolysis, just to make sure we understand the using and gaining energy. So as we go from glucose to pyruvate, okay, we use 2 ATP right in the beginning there, in the first and third step. All right, we burned it up. Okay, we are gaining along the way. We convert 4 ADPs to 4 ATPs. All right, we get 2 piggy banks out of it. We convert 2 NAD pluses to 2 NADHs. So when all is said and done, our net gain is 2 ATP and 2 NADHs. So even though we made 4 ATP, we used 2 ATP. So the only real advantage we get converting glucose to pyruvate is 2 ATP and 2 NADH. So we have some minor energy investment of 2 ATP and a small energy return. So again, glycolysis, and that's how bacteria learn to metabolize sugar, really didn't give a ton of energy. All right, so 1 6-carbon sugar, we're left with 2 3-carbon sugars. All right, pyruvate, though, now this is not the end of the process. This is just glycolysis. Pyruvate has a lot of energy in it that has not been captured. It still has 3 carbons bonded together, and we've got energy in those bonds, and it can be oxidized further. Okay, but glycolysis ends at pyruvate. All that work, and that's all I get. And that's what I always think when I look at glycolysis, and I'm sure you're thinking that too. Really complex number of steps to just get a little bit of ATP. Okay? And glucose has a lot more to give, right? All we did was take a 6-carbon sugar and turn it into 3-carbon sugar, and we got just a tiny bit of ATP out of it. But now we can take those 2 3-carbon sugars and get more. Okay? It's not a lot of energy, but for 1 billion years, this is how life on Earth survived. There's no oxygen, so there's slow growth, slow reproduction, and we're only harvesting a tiny bit, 3.5% of the potential energy in glucose. All right, we have a bunch more carbons to get off of there and more energy to find. All right, but those early bacteria had no oxygen, so they could only do the part glucose to pyruvate that doesn't require any oxygen. All right, so why does glycolysis still take place? Well, it's still an efficient way to break down glucose, and we can take those pyruvates and do something else with them. Right, there's that basic second half. All right, raw materials to products. Glycolysis, okay, here's the whole formula of glycolysis. Glucose plus the 2 ATP, essentially, that we're going to use, 2 ADP and 2 phosphate groups and 2 NAD pluses, we're going to yield 2 pyruvate, 2 ATP, and 2 NADHs. All right, so they're showing you that. All right, we're going to run out of NAD+. Okay, without regenerating NAD+, energy production would stop. Another molecule must accept the hydrogen from NADH so that NAD plus is freed up for another round. So how are we going to recycle? Okay, so how is NADH recycled to NAD plus? Okay, another molecule must accept hydrogen from NADH. So that kind of brings us to our next steps in the process of cellular respiration. Okay, how are we going to recycle the NADH? Alright, so there's our pyruvate molecule. And if you've got oxygen, alright, you're going to go through aerobic respiration. And you're going to go through the rest of cellular respiration and the Krebs cycle, and you're going to recycle your NADH, and you're going to get a whole bunch more energy, and we'll talk about that in the next video. And without oxygen, okay, you're going to go through anaerobic respiration, and there's two kinds of anaerobic respiration. We're going to talk about fermentation. So we have two types, the lactic acid fermentation and alcohol fermentation. Right, so lactic acid fermentation is what your muscles do when they run out of oxygen, and alcohol fermentation is what organisms like yeast do. Alright, so that's how you can recycle NADH if you don't have any oxygen. It's fermentation. Alright, so if you've got oxygen, right, like a nice healthy human being running around after she just ate a bunch, okay, then you're going to convert pyruvate and go through the Krebs cycle, get a bunch of energy, and recycle NADH. If you don't have any oxygen and you're a muscle, you're going to do lactic acid fermentation, or if you're a little single-celled yeast critter, you're going to go through alcoholic fermentation. Okay, so which path you use depends on who you are. So fermentation is done by bacteria and yeast. So we take pyruvate, that 3-carbon sugar, and convert it to ethanol and carbon dioxide. All right, and where's that NADH come in? Well, right there, okay, it gets recycled, all right, and can go back to glycolysis. Okay, so that's how we make beer, wine, and bread, is by taking advantage of fermentation. Okay, so when you are eating bread and you see all those holes that are in bread, that's the gas, the carbon dioxide that comes off of the yeast after it's eaten the sugar. Okay, animals and some fungi do lactic acid fermentation, where we convert pyruvate to lactic acid, and we recycle our NADH back into NAD plus and back to glycolysis. And that's how we make cheese, okay? And that's how you can continue to get your muscles to work in the absence of oxygen. So if you're sprinting a race, you could run out of oxygen in your muscles, but your muscles you want to keep pushing them past that point of exhaustion. Your muscles are going to do some lactic acid fermentation. You've probably heard of lactic acid buildup in muscles. So if you're a weightlifter or a serious athlete and you're constantly pushing your muscles past the point of aerobic respiration, you end up with some lactic acid residuals in your muscles that can kind of make you more sore. Lactic acid, though, is not really like ethanol where it's just done. Once you have oxygen again, you can take the lactate and convert it back to pyruvate using your liver and then go on to the Krebs cycle. Right, so here's alcoholic fermentation done by bacteria and yeast. Pyruvate into ethanol and carbon dioxide. Excuse me. Okay, it's a dead-end process. Okay, around 12% ethanol and kills the yeast, and you can't reverse the reaction. Okay, count the carbons. So you want to see that we're ending up with the same number of carbons that we started with. So we have 3-carbon pyruvate. Ethanol has 2 carbons in it, and carbon dioxide has 1 carbon in it. So So we have 3 carbons on each side, so it's a balanced reaction, right? And so as you see, we go from glucose to the pyruvate still. That part of glycolysis is still there. We recycle the NAD plus, the NADH rather. Okay, and we end up with ethanol and carbon dioxide. Lactic acid fermentation, done by animals and some fungi, converting pyruvate to lactic acid, 3 carbons on each side, converting NADH to NAD plus in the process. Okay, the NAD plus can go back to glycolysis to help out the steps of glycolysis. Now this one is reversible. Okay, we counted the carbons already. All right. Once oxygen is available again, lactate can be converted back to pyruvate by the liver. Okay, pyruvate is a branching point. All right, without oxygen, fermentation happens. Okay, and anaerobic respiration to get a little bit more energy and to recycle our NADH. Okay, with oxygen, we're going to go to the mitochondria. That pyruvate is going to the mitochondria to go through the Krebs cycle. And that will be the rest of aerobic respiration, which will be in the next video. Okay, what's the point of all this? The point is to make ATP, of course. And that ends part 2. I hope this was helpful.